There are four kinds of structures in any polypeptide: primary structure, secondary structure, tertiary structure, and quaternary structure. The secondary structure results from hydrogen bonds between backbones of amino acid sections in the macromolecule. These orange lines show the hydrogen bonds between the backbone of two strands of a single polypeptide. The tertiary structure of a polypeptide results from bonds between the R groups of individual amino acids in the long polypeptide chain. This causes folding structures more complicated than the simple adjacent structure resulting from secondary characteristics. Tertiary bonds are indicated here in green. The quaternary structure of a protein results from the way that different sections or polypeptides join all together to form a fully functional protein. The bonds connecting different sections of a fully functional protein are indicated here in red. Recall that all 20 amino acids can always be found in a fully functional protein. On the bottom left of page 40, we've drawn some proteins associating with a lipid bilayer in cross-section. There is polar aqueous solution to the exterior of the cell, called interstitial fluid, and polar aqueous solution to the interior of the cell, which is called cytoplasm. Recall our general solution rules. Like dissolves like, or polar dissolves polar, and nonpolar dissolves nonpolar, and nothing large and nothing charged enters or exits the cell without the help of a protein. Amino acids can be nonpolar, polar, or electrically charged depending on the R groups attached to them. The nonpolar amino acids are glycine, alanine, valine, leucine, isoleucine, methionine, phenylalanine, tryptophan, and proline. The polar amino acids are serine, threonine, cysteine, tyrosine, aspartagine, and glutamine. The charged amino acids are aspartic acid, glutamic acid, lysine, histidine, and arginine. This chart shows the symbols that stand for each of these individual amino acids. If we apply the like dissolves like rule to amino acids, we can determine where an amino acid would likely be found in a fully functional protein based on the polarity of the things that it's in contact with. Polar amino acids would likely be in contact with either of the aqueous solutions on both sides of the lipid bilayer, the polar heads of the lipid bilayer, or potentially the interior of the channel going in and out of the lipid bilayer, since it would be in contact with aqueous solution. We would likely find nonpolar amino acids touching the nonpolar tails or embedded in the interior of a peripheral protein like this. Electrically charged amino acids could be in touch with the aqueous solution in any area, or if this ion channel's purpose is to move charged particles into and out of the cell, you would certainly expect electrically charged amino acids to line the channel. They would be able to grab a charged particle and then, based on their pK values, hand them to a neighbor electrically charged R group down the chain as the pH or charge of the interior of the channel changes in reaction to the movement of ions. I've included an easier to read scanned copy of the notes under downloads on the website for the class.